Hello friends, today we are going to study about protein function, that is reversible binding of a protein to a ligand, part 5. Hemoglobin transport hydrogen and carbon dioxide also. Till now we are very much familiar with the function of the hemoglobin. What is the function of the hemoglobin? It carries oxygen from lungs to tissues. But apart from carrying oxygen, hemoglobin also carries H plus and CO2. What are these? These are the end products of respiration. Now, we are already familiar what is respiration? That is breakdown of food in the presence of oxygen to release H2O and CO2. Now, this CO2 will react with water to produce a bicarbonate. The CO2 generated during breakdown of food will react with water to form hydrogen ion and HCO3 minus, that is bicarbonate. And the enzyme required for this process is carbonic anhydrase. This enzyme is abundant in erythrocyte. Now, why this conversion is needed? First of all, carbon dioxide is insoluble in aqueous solvent, due to which what happens is it will form bubble in blood and tissue if not get converted. And the second thing needed is while conversion, H plus is released. So more of H plus means less pH. So this pH and the carbon dioxide concentration, or we can say the H plus concentration and the carbon dioxide concentration affects the binding affinity of oxygen with hemoglobin. We all know hemoglobin carries oxygen. Now it is important to release the oxygen in the tissues. And how it can release the oxygen? We already studied that the partial pressure of oxygen in arteries is 100 torr and in veins it is 30 torr. So hemoglobin is fully saturated in arteries and half saturated in veins. And this difference is important to release oxygen in the muscle tissues. Now, apart from these difference, two other things plays an important role for the release of oxygen in the tissues. And what are these things? Hydrogen ion concentration and carbon dioxide concentration. So therefore, the conversion is important. First, because carbon dioxide is insoluble and form bubbles in blood. And second, for the pH. So carbon dioxide is not soluble in water and it releases H plus, which is needed to release oxygen by hemoglobin in the tissues. We'll see how it happens. Now, hemoglobin transport 40% of total H plus and 15 to 20% of CO2. What about rest H plus? The 60% is absorbed by plasma bicarbonate buffer. And what about carbon dioxide? So these are transported as dissolved HCO3 minus and CO2. The binding of H plus and CO2 is inversely related to the binding of oxygen. That means wherever the concentration of H plus and CO2 will be high, the hemoglobin will bind with H plus and CO2. To bind with H plus and CO2, it will release the oxygen. And when hemoglobin will bind with oxygen, it will release H plus and CO2. So what happens in peripheral tissue? Because of respiration, H plus concentration is high in the tissue and CO2 concentrations are also high in the tissue. So that means in the tissue, the hemoglobin and oxygen affinity will reduce. That means what will happen? It will release oxygen in the tissue and it will bind H plus and CO2. What happened in the lungs? Reverse. In the lung, H plus concentration is low, CO2 concentration is low. That means hemoglobin binding affinity with oxygen will be high. That means hemoglobin will bind with oxygen and will release CO2 and H plus. So now this effect of pH means H plus and CO2 concentration on the binding and release of oxygen by hemoglobin is known as Bohr effect. Now, when the hemoglobin binds with oxygen, it forms HBO. This is the binding of one molecule of oxygen with hemoglobin. Now, to account for effect of H plus, if we want to know the effect of H plus concentration on this equilibrium, the reaction will become like this. What this reaction is saying? When the hemoglobin is bound with H plus, it released O2. When the hemoglobin is bound with O2, it released H plus. That means they are inversely related. The binding of O2 and H plus is inversely related to each other. The same is the situation with CO2 as well. So both oxygen and H plus are bound by hemoglobin, but with inverse affinity, and the same is true for O2 and CO2 as well. But the oxygen and H plus, they don't bind at the same site. Where oxygen binds with Fe2 plus of heme, where H plus bind, it binds with the side chain of amino acid residues. Now the Bohr effect is mediated by histidine 146. What happens is, whenever H plus concentration is more, this H plus will go and bind with histidine 146. Because of which this histidine became protonated, and this protonated histidine form ion pair with aspartate 94. And what we studied? The ion pair stabilizes T state. T state means deoxyhemoglobin state. So, what is happening at the molecular level? More of H plus in the peripheral tissue means it will protonate the histidine 146, which will form the ion pair with aspartate 94, stabilizing the T state, that is deoxyhemoglobin state. That means in the peripheral tissue, the hemoglobin will release oxygen. What happened in the lungs? H plus is less, so no protonation. No protonation means no ion pair. No ion pair means R state is stabilized. That means they will release H plus and CO2 and bind oxygen. So this is the condition in lungs. The above one is the condition in peripheral tissues. What is the pH in lungs? 7.6. What is the pH in tissues? 7.2. So less pH means more of H plus. More of H plus means protonation, ion pair formation, and T state stabilization. More pH means less of H plus. Less H plus means no protonation, no ion pair, and R state will be stabilized. That means it will bind oxygen and release carbon dioxide and H plus. The same thing has been plotted here in the graph. As you can see, as the pH is decreasing from 7.6, 7.4, 7.2, the graph shifts towards the right hand side. That means less pH means H plus is more. So here oxygen binding will be less, and hence the graph shifted towards right side. So thus, four polypeptide chain of hemoglobin, they communicate with each other not only for oxygen binding as well as for the hydrogen binding. Now hemoglobin also binds with carbon dioxide in the same manner as it does with the hydrogen ion. It forms carbamate group to the alpha amino group at the amino terminal end of each globin chain, forming carbinohemoglobin. Now this is in short about the Bohr effect. What it is happening in the lungs, when the partial pressure is more, it will bind with the hemoglobin. This oxyhemoglobin will transport it via blood to the muscle tissue. In the tissue, the hemoglobin will release oxygen. This oxygen will be picked up by the myoglobin. It will release the oxygen in the muscle tissue where oxygen will be used for the respiration purpose. As a byproduct of respiration, carbon dioxide will be formed. This carbon dioxide will react with water to form bicarbonate and H plus. This H plus will also transported via hemoglobin, and this bicarbonate will again, in the presence of enzyme carbonic anhydrase, will convert into CO2 in the lungs to be exhaled out. So this is the Bohr effect, that is the effect of H plus concentration and CO2 concentration on binding affinity of oxygen with hemoglobin. Here in place of H plus, you can also say it as a pH, effect of pH and carbon dioxide. Binding of hemoglobin with oxygen is also regulated by 2,3-biphosphoglycerate. The relation of 2,3-biphosphoglycerate is same like H plus and CO2, that is inversely related. So when BPG will be there, the binding affinity of hemoglobin with oxygen will be reduced. This can be given with this equation, that is hemoglobin when bind with BPG, it will release oxygen. Hemoglobin when bind with oxygen, it will release BPG. So the effect of BPG is same like H plus and CO2, that is inversely related to the binding affinity of oxygen with hemoglobin. Now BPG binds at different position in hemoglobin. Where it binds? It binds inside the cavity between beta subunits. It stabilizes T state because its binding releases oxygen. Now BPG is negatively charged, therefore it can bind with the positively charged cavity of hemoglobin. So BPG and binding affinity of hemoglobin with oxygen, these are inversely related. Now this BPG also helps in adaptation with higher altitude where oxygen concentration is less. How? As we already studied, that partial pressure in arterial blood is 100 torr and in venous blood is 30 torr. Now, this difference is needed to release oxygen in peripheral tissues. Now, at higher altitude, where oxygen concentration is less, there this arterial pressure won't remain 100 torr. It will reduce. So, when it will reduce, this difference will also reduce. And because of this, the release of oxygen in tissue will become difficult. So how to solve this problem? In higher altitude, the concentration of BPG increases in blood. So more of BPG means less of binding affinity of oxygen with hemoglobin. That means when BPG concentration is more, the hemoglobin will release oxygen. So that's how BPG helps to release oxygen even at when the oxygen concentration is not 100 torr or even at when the oxygen concentration is reduced at higher altitude. So whenever we go at higher altitude, our body gets adapted by increasing the level of BPG in the blood so that hemoglobin can deliver efficiently oxygen in our peripheral tissues. Now, this BPG also have a role in fetal development. Now, how that happens? Fetus take blood from mother body. So that means fetus has to extract oxygen from mother body. To extract oxygen from the mother body, it should have very high affinity of hemoglobin with oxygen compared to maternal hemoglobin. So the affinity of hemoglobin with oxygen should be much more than the maternal hemoglobin because they are extracting from the mother body. So in fetus, the BPG concentration is low so that hemoglobin binding affinity with oxygen should be more. And for this function to perform, the beta subunit of hemoglobin in fetus get replaced with gamma subunit. That means in fetus, we don't have alpha 2 beta 2 subunit. What we have is alpha 2 gamma 2 subunit. And this gamma 2 has lower affinity for BPG and due to which it can bind with more affinity with oxygen. Clear? So that's how BPG helps in fetal development also, and BPG helps in adaptation at higher altitude also. Now, one question from my end for you all. What is the effect of following changes on oxygen affinity of myoglobin and hemoglobin? A drop in the pH of blood plasma from 7.4 to 7.2, a decrease in the partial pressure of carbon dioxide in the lungs from 6 to 2, an increase in the BPG level from 5 to 8. Do let me know in the comment section if you found the video informative. Please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.